all of my data, I wanted to point out and kind of like preface my talk and point out that a lot of the data I'm going to be talking about today was either collected by undergrads that I've worked with and trained, or with the assistance of those undergrads. So most of the experiments that I'm going to be talking about today, any of you could learn how to do with a couple of weeks of training. All right, so I've already got a little bit of background on what tardigrades are. So here is a color-enhanced image of a scanning electron microscope image of a tardigrade hanging out on a piece of moss, also called water bears and moss piglets. And most people that I talk to have, if they've heard of them, they've heard about them from Neil deGrasse Tyson talking about them on Cosmos, or Bill Nye talking about them on Bill Nye Saves the World, but apparently Octonauts is also out there. I'm going to have to look that up. Yeah, that's going to have to go on the list too. So these are microscopic animals, and they're less than 1 millimeter in length. And you can see one here in comparison to a C. elegans roundworm, in case that's a useful point of comparison. And our lab is interested in tardigrades because of their ability to survive this wide range of extreme conditions, including almost absolute zero, above the boiling point of water, vacuum, 6 times the pressure at the deepest part of the ocean, 1,000 times more radiation than the typical animal, at least 10 days exposed to the vacuum of outer space and radiation from the sun, at least 10 years desiccated. Desiccation is basically dehydration, but to an extreme. So they can survive at least 10 years desiccated to as little as 1% water. And they can survive at least 30 years frozen. However, for the conditions that are bolded here, tardigrades first have to enter into this hibernation state, dehydrated hibernation state, and it takes them about 24 hours to enter into that hibernation state. And if they dehydrate too quickly during that process, they die. And a postdoc in our lab, Thomas Boothby, looked at how tardigrades take this time to enter into the hibernation state and came up with this hypothesis that they were using that time to synthesize molecules to protect their cellular components against those extreme conditions, particularly desiccation and heat. And what those molecules are and how they work could potentially have a lot of impact on how we stabilize protein-based drugs. So to give you an idea of the scope of this problem of stabilizing drugs, let's take a look at Dalal, Ethiopia, which is the hottest inhabited place on the planet. It has an average annual temperature of 35 degrees Celsius, which translates to 95 degrees Fahrenheit. And that's the average annual temperature, so the temperature is often spiking above that. In contrast, most or many drugs have to be stored under refrigeration. And there are a lot of places in the world where that can't be done because they don't have electricity or it's too expensive. Drugs, or just in very rural, remote areas, that's difficult to do. All right, and this affects a wide range of medications, including vaccines, insulin, antivenoms, and an increasing number of monoclonal antibody-based drugs, which you've probably seen commercials for, and I've included a couple of them down here. And these protein-based drugs are becoming a larger and increasingly larger share of the market for medications. In 2012, worldwide, people spent $87 billion on protein-based drugs. In 2016, that was up to $192 billion. And it's projected that by 2020, people will be spending $400 billion. So there's also this, in addition to there being an opportunity to make drugs more accessible to people, there's an opportunity to make them more affordable for people, and for the pharmaceutical industry to make money in that process. All right, now there are molecules out there that are currently used to stabilize protein-based drugs. And so we've been using a couple of these as controls, seeing if we can find something in tardigrades that's more effective than them. So one example is trehalose, another is serum albumin, and I'm going to go into a little more detail about what these molecules are and where they come from, since they'll be popping up throughout our experiments. All right, so trehalose is a non-reducing sugar, which means that this bond between the two monosaccharides is difficult to break. So it's more stable than the average sugar. And trehalose is synthesized by a lot of desiccation-tolerant organisms, including brine shrimp, which are also known as sea monkeys, yeast, plant seeds, and in some plants. And trehalose is also made by a lot of fungi, which is why it's very easy to cook with dehydrated mushrooms and they just kind of like spring back. And trehalose for decades has been FDA approved in numerous protein-based drugs. So it's a really good control for our experiments. All right. The other excipient that we've been using as a control is serum albumin. Human serum albumin is the most abundant protein in blood plasma. And it's FDA approved in formulations with 3 different drugs, with a dozen more in the clinical trials process. So it's a much newer excipient on the market, but it's increasingly being FDA approved. And conveniently for us, it is homologous to bovine serum albumin, or BSA, which is a byproduct of the meat industry, so we can buy a lot of that protein for very cheap. So we've been using BSA as a stand-in for human serum albumin in our experiments. You can see that they have very similar folds. There's the overlay of the 2 proteins, and they're 71% sequence identical. All right, so you may be asking, why don't tardigrades just use these molecules? We know that the species of tardigrades that we've been focusing on, they aren't making the enzymes needed to synthesize trehalose. So tardigrade, at least these tardigrades aren't making trehalose. And tardigrades also don't have a bloodstream, so they're not making serum albumin. They don't have the gene for serum albumin, so they've got to be making something else. And that's something else that we've been studying is the cytosolic abundant heat-soluble protein family, or CAHS proteins. And these proteins are upregulated 4 to 22-fold under dehydration stress. So in other words, when tardigrades are under dehydration stress, they're making up to 22 times the amount of messenger or RNA corresponding to these proteins. And this family of proteins is unique to tardigrades and has not been found in any other species whose genome has been sequenced. So that kind of gave us an idea that between those 2 things, that this could be how tardigrades are surviving desiccation. To break down what the name of this protein means, you'll want to remember the cytosol. So that's going to be the orange space in the cell diagram, all of that area in between all of the organelles. And so a cytosolic abundant protein is just abundant in the cytosol, which is not uncommon. There are plenty of proteins that are abundant in the cytosol. What's more unique about these proteins is that they're heat soluble. Most proteins, if you heat them up, they cook like in an egg. And the CAHS proteins from tardigrades don't do that. And so that's kind of what makes them one of the initial things that we knew that made them special. To understand how they're heat soluble, we have to kind of look at their protein structure. So to review that, looking at the primary structure of a protein, that's the amino acid sequence, we have that folds up to form secondary structure elements, alpha helices and beta strands. Those fold up further into tertiary structure. And then folded proteins come together to form protein-protein interactions in quaternary structure. And there's an additional type of structure that's less discussed, but becoming, we're realizing it's increasingly that it's important, and that's quinary structure. So if we zoom into the inside of a cell and look at that environment, quinary structure is all of those transient interactions that occur between different proteins in the crowded environment of the cell. And so you can see within this simulation, we've got a ribosome in gray on the left. That's that biggest protein. There are some transfer RNAs, tRNAs floating around, and a bunch of other different proteins all crammed together. Now you can imagine that if you heat up this crowded environment of proteins, the protein-protein interactions are going to start to fall apart. The tertiary structure is going to start to fall apart. And instead of refolding properly, a lot of the times those alpha helices or beta strands are going to grab onto a neighboring protein that they normally wouldn't interact with. And that's how you end up with this kind of like gelatinous cooked egg effect. All right, so our tardigrade proteins, our CAHS proteins, are able to avoid this through a property known as intrinsic disorder. So globular proteins, which you're more likely to be familiar with, those are these proteins composed of alpha helices and beta sheets. You can see in the globular region of this protein that there is movement within the protein, but overall it maintains the same general structure. There are also disordered regions of proteins and intrinsically disordered proteins. And these sequences are highly flexible, highly dynamic, constantly moving around. They don't have a consistent structure. And our CAHS protein is a good example of that. So here's a computational model of what our CAHS protein might look like in solution. And you can see that it's like that highly dynamic, constantly moving, intrinsically disordered protein. Because of that, there's not much to unfold in this protein, and there's not much to misfold or bind to neighboring proteins, which is how this protein is heat soluble. All right. So these properties of being intrinsically disordered and heat soluble, those are also properties of other proteins that have been linked to desiccation tolerance in other species. So that was another indicator that this family of proteins might be used by tardigrades to survive desiccation. And so at this point, we went on to directly test that in tardigrades. All right, so we used a technique called RNA interference. So for this technique, we interfered with translation of the messenger RNA into various CAHS proteins. Those CAHS proteins are down here on the x-axis. We also looked at interference for GFP, the green fluorescent protein, as a control because tardigrades don't have that in their genome and therefore don't make that. So that just was a control to make sure that the technique itself that we were using wasn't messing with tardigrade survival. So we looked at inhibition of translation of these different proteins and how that affected the survival rate of tardigrades under hydrated and dehydrated conditions. Under hydrated conditions, if you inhibit translation of these proteins, there is no really real effect on the survival rate, at least none that is statistically significant. However, if you take those tardigrades and dehydrate them, the tardigrades in which translation of CAHS proteins has been inhibited are much less likely to survive under dehydration stress. So from that, we're able to conclude that tardigrades need these CAHS proteins to survive desiccation. We can also trick several other species into making these CAHS proteins and seeing, we could then see does making those proteins increase the survival rate of those species? So first we did that with E. coli, and we measured the desiccation tolerance of E. coli, and you can see this is a logarithmic scale, and how that was affected by expression of various CAHS proteins. And here we have alpha-synuclein, an intrinsically disordered protein control, just to make sure that it wasn't a general property of that class of proteins. And it varies depending on what CAHS protein you look at, but expressing these proteins in E. coli allows you to increase the desiccation tolerance by over 100-fold for E. coli. We've also run this experiment in yeast and saw a very similar effect. Again, this desiccation tolerance is on a logarithmic scale, and again, we see up to 100-fold increase in the desiccation tolerance of yeast when you express these CAHS proteins. So that told us that we had this really cool family of proteins to look at, and how it might be, we might be able to relate it to stabilizing drugs. If you want to know more about our work with tardigrades themselves, you can find it in this paper that we published earlier in the year. But I'm going to jump into our experiments that looked at how we might be able to stabilize different types of enzymes and proteins in vitro. So first, we started by looking at the ability of this family of CAHS proteins to protect an enzyme against desiccation with and without heat. We've investigated the gelation properties of this family of proteins and how another protein encapsulated in that gel is stabilized by the CAHS gel. We've started probing the structure of that gel as well, using circular dichroism, some computational modeling, and we've, from that, have a potential mechanism of gelation. All right, so to start off with, I'm going to go into our data looking at the ability of these proteins to protect an enzyme against desiccation and heat, which tells us both how tardigrades are surviving those extremes, and also could we use these proteins to stabilize dehydrated formulations of protein-based drugs. All right, so we're drawing our proteins from 2 different species of tardigrades, each of which has over a dozen genes for CAHS proteins. And there are 1,200 species of tardigrades that have been documented. So we're looking at a very small subset of the possible sequences out there in this family of proteins. A subset of the CAHS genes are upregulated under dehydration stress. That means that they're making a lot more messenger RNA for these genes corresponding to these genes under dehydration stress. And we selected 5 to focus on. We put these genes in plasmids, and then we have E. coli synthesize the protein for us, and we can purify the protein from E. coli. All right, and we tested the ability of these proteins to protect the enzyme lactate dehydrogenase. Okay, here's the crystal structure of that protein, and it converts pyruvate into lactate following this arrow pushing mechanism with NADH donating a hydride group. We're able to observe the conversion of NADH to NAD+, and through that infer the enzyme activity using a UV-vis spectrophotometer and looking at the absorbance of our solution. NADH absorbs light at 340 nanometers, and NAD+ does not. So as a result, if we have our enzyme in solution with reactants, assuming that we have a surplus of reactants, we can get the rate of the zero-order reaction of that enzyme as the absorbance changes over time. And that tells us how active the enzyme is. All right, so how this experiment looks, we take a sample of enzyme in solution, take half of it, put it in the fridge, store it, and that is our 100% activity control. That's how we know how much activity there was initially. We then remove all of the water using a speed vac, which is a centrifuge hooked up to a vacuum pump. That leaves us with a dehydrated protein mixture. And then we add water back in. And we do this in the presence of various CAHS proteins and control molecules. All right. So we're able to take our stressed sample, compare it to that sample we saved in the fridge at the beginning of this experiment, and determine the percent activity remaining. And we looked at that over a wide concentration of excipients. All right. So lactate dehydrogenase, or LDH, on its own with nothing there to protect it, it really should be at 0, but since we're at a logarithmic scale on this axis, there is no 0. So I've placed it at 0.1, but you can see that without anything to protect it, less than 1% of the enzyme activity is remaining. All right, now if we add in trehalose, trehalose is able to protect the enzyme. So throughout this data set, notice that there are error bars. So we got those error bars from running the experiment 3 times. And we've also fit this data with a red curve, which is not for any mathematically significant reason, just because it's visually easier to look at the data that way. If we compare that to the ability of BSA to protect LDH, BSA is better. And our tardigrade proteins, the 2 that we've looked at, both exceed these known excipients. All right, and we can quantify how much better they are by looking at the percent activity, 50% activity, and the amount of excipient required to protect 50% of the enzyme. Okay, so if we look at this in terms of grams per liter, BSA is about twice as effective as trehalose, but our tardigrade proteins are an order of magnitude more effective. If we convert everything to mole ratios, you need a lot of trehalose to protect LDH. That's because trehalose is such a small molecule compared to the proteins. The proteins dwarf that disaccharide. However, our CAHS proteins are still 2 to 4 times better than BSA at protecting if we look at things in that context. All right, so all of those experiments were collected at room temperature. We also wondered, how is heat affecting protection? So for this experiment, it's very similar. Vacuum off the water, we're left with this dehydrated mixture. We heat that up to 95 degrees Celsius for various amounts of time. So that's almost the boiling point of water. Cool it back down to room temperature and add water back in. And again, look at the LDH activity. For these experiments, we used a consistent excipient concentration at 20 grams per liter. So at 0 minutes, the activity will fall along this line. All right, so trehalose has almost no ability to survive this extreme heat. We see almost complete loss of activity in 5 minutes. BSA does much better. However, we still lose almost all activity in 4 hours. Our tardigrade proteins, on the other hand, we see activity, we don't see a drop in activity until after an hour at 95 degrees Celsius, and then we still see some activity after 24 hours. So this data is very promising. It suggests that we could potentially use these CAHS proteins from tardigrades to stabilize dehydrated formulations of biologics and help solve this refrigeration problem worldwide. All right, so after collecting all of that data, we started wondering, how are the proteins protecting this enzyme? How does that work? And an obvious potential answer to that question came during our purification process, and that's that these proteins gel. And they do that very much like Jell-O. It's a completely reversible process that's concentration dependent, temperature dependent, and time dependent. And this is such a prevalent property that if you take E. coli cells that are overexpressing these proteins, suspend them in as little volume of buffer as possible, and lyse them by sonication, so burst them with sound waves, and flip your tubes upside down, that kind of like cell guts mixture immediately turns into a gel. So you can see our 5 different CAHS proteins and alpha-synuclein and intrinsically disordered protein control does not have that property. These samples are blue just because we added some dye to make it easier to photograph. So that has nothing to do with the actual experiment. And so this was really cool and really weird. We did not expect to see this at all. But this was a very qualitative observation. We wanted to concretely demonstrate that gelation is occurring. And so for that, we've been using a technique called Rheology, which is the study of the flow of matter. And it's often used to study viscoelastic materials like gels. So for this type of experiment, we applied an oscillating strain to one side of our sample and measured the stress response on the other side. For a perfectly elastic solid material, the output stress is going to be in phase with the input strain. For a perfectly viscous fluid, the output stress is 90 degrees offset from the input strain. And for a viscoelastic material that has a combination of solid and liquid properties, like a gel, we see a mixture of these two. We can deconvolute that using this formula and some trigonometry and determine how elastic or how solid the sample is in terms of a storage modulus, and how viscous or how liquid the sample is in terms of the loss modulus. All right, so here's the data we get. Storage and loss modulus on the y-axis. And for this experiment, we're looking at a sample of our CAHS protein as it gels over time. While the loss modulus is greater than the storage modulus, the sample is more liquid than solid. While the storage modulus is greater than the loss modulus, the sample is more solid than liquid. And where those two curves cross over is the point of gelation. So that's where it turns from a liquid into a gel. All right, so we can use this type of experiment to determine how, or what the concentration dependence of gelation is, what the salt dependence is, temperature dependence, all of those things. And we've begun preliminary experiments to do that. We can also look at these gels using a scanning electron microscope. So you can see some examples of those images here. And you can see that this is a very porous material. And that led us to a more refined hypothesis, that our CAHS proteins were encapsulating and protecting other proteins and enzymes in that gel, and protecting them against desiccation and heat. And this was a hypothesis that we could directly test. All right, and it also tells us, can we use these proteins to stabilize hydrated formulations of protein-based drugs? All right, so for these experiments, instead of lactate dehydrogenase, we're using the test protein SH3. SH3 stands for N-terminal SH3 domain of Drosophila signal transduction protein, DRK. But we're using this protein not because of anything really related to the name of it, but because it's metastable. So what that means is that about half the time, it's nicely folded like this, and the other half the time, it's unfolded, and we see exchange between these two states. And we can study this protein using nuclear magnetic resonance spectroscopy, or NMR. So those of you who've taken organic chemistry, think back to what you learned in that class, although I'm hoping that I can explain it well enough that everyone will be able to follow this. So NMR allows us to study specific types specific isotopes of atoms. So the ones that are most commonly found in a protein, protons, carbon, nitrogen, oxygen, we could use NMR to study those atoms. However, there are hundreds of each of those in any one protein. We're able to greatly simplify our NMR experiments by instead using fluorine. So fluorine is rarely found in nature, rarely found in proteins, and not found in this particular protein. And we can incorporate it into the single tryptophan in SH3. We can basically trick E. coli into putting a fluorine atom in this position. All right. And so this allows us to get a very simplified spectrum where we have one peak corresponding to the population of the protein in the folded state, and a second peak corresponding to the population of the protein in the unfolded state. So if our encapsulation in our CAHS protein stabilizes SH3, we would expect to see a shift and an increase in this peak, the folded peak. All right. So, and we did see that. So here's that data. You can see in black, we have SH3 in buffer, and then in blue, we have SH3 encapsulated in our CAHS protein gel. And there's an increase in the folded peak when we encapsulate SH3 in the gel. We can integrate over those peaks and quantify the free energy of unfolding. So if this math looks intimidating, you've probably seen something similar in your basic chemistry class. It's just a couple other variables fit in this free energy equation. All right. Okay. And so we can calculate the free energy of unfolding of SH3 over a range of CAHS protein concentrations. And you'll see at this highest concentration at 35 grams per liter, we have this sudden increase in stability of this protein. And this corresponds to what we've seen in our preliminary rheology experiments as the point of gelation. So we need to flesh out this data a little bit more, run replicates of it to make sure it's reproducible, but it's very promising and suggests that we could potentially use CAHS proteins to stabilize hydrated formulations of protein-based drugs. Okay, so we showed that this gel can encapsulate and stabilize other proteins. Next question we asked was, how does that gel form? And so this is something that we have a lot more experiments to run to fully figure out, but I'm going to show you some of our preliminary data. So first, we've been using circular dichroism spectrapolarimetry to study the secondary structure of our CAHS proteins in the solution state and in the gel state. All right, for the sake of time, I'm not going to get into the nitty-gritty of the physics of how this works, but there's something called circularly polarized light, and we can shine that through a protein solution or a protein gel. And each secondary structure element has a characteristic spectrum that results. And so for a protein like this that has several different secondary structure elements, you'll see these different spectra add together, and you can deconvolute them to figure out percentages of how much alpha helix is there, how much beta sheet, how much disordered region is there. Okay. All right, so here is some of our preliminary data. So in solution, our CAHS protein is largely disordered. You can see that there's some alpha helix and some beta sheet here. We know from experiments that we've previously published that all of this structure is transient. So it may show up briefly and it may be visible by circular dichroism, but it also falls apart briefly, or as soon as it forms, it very quickly falls apart. So it's not a very stable structure. Now, if we concentrate that protein and it gels, we see this large shift to alpha helical content. So that suggests that the gel is formed in large part by alpha helices, although we're not exactly sure what that looks like yet. All right. So if you remember this computational model of our protein from earlier, we're going to take a closer look at it as well. So if you look at this protein, you can see that there's an extended linker region and that the termini are more collapsed in structure. They're not consistently alpha helical, they're not consistently beta sheet, but they're collapsed and they're not extended like the center region. And we can see that throughout the simulation if we run it again. All right. And so from that, combined with our circular dichroism data, we have a potential mechanism of gelation. So we have our CAHS protein with more structured termini separated by an elongated linker region. And in dilute solution, those proteins are probably moving very similarly to that computational model I just showed you. However, if you concentrate that protein or cool it down so that it gels, those termini probably come together to form that gel network. This allows for the reversibility that we've seen in our experiments. And it gives us a mechanism that we can very concretely test in the lab. So that's kind of our future direction of where we're going. So we're going to try and figure out exactly how this gel forms and how that protects other proteins. All right. So in summary, we've got this really cool family of proteins from tardigrades that can protect enzymes against complete dehydration, complete desiccation, and extreme heat. These proteins form gels, which is an unusual property for any protein. That gel can stabilize other proteins encapsulated within it, and we're starting to look into how that gel forms. All right, so I would like to thank my lab, particularly my boss, Gary Pilak, who has been kind enough to let me use his office for this webinar, postdoc Thomas Boothby, who has really laid the groundwork in terms of the biology side of things for my project. Grad students on our team Tardigrade, Candace, Shannon, and Alex. And then as I mentioned at the beginning of the talk, we have a bunch of undergrads that work on this project. So we have Akash, Kenny, and Cody. And then we even have a high school student, Ashley, whose data I did not talk about today, but who collected some great LDH enzyme assay data for us over the summer. We have another half of the lab that looks at protein crowding and how that affects protein stability, folding, interactions. And we have fantastic collaborators, Professor Bob Goldstein at UNC, who was the first person to tell us about tardigrades, Sergei Shako's lab, who has taught us about rheology, Rohit Papu at WashU, who did the computational modeling for us, and then Bonnie Wallace at University of London, who is helping us run our circular dichroism experiments. And of course, the National Science Foundation for giving us money, without which we wouldn't be able to run any of our experiments. Okay. UNC is a great place to do research, and there are a lot of research opportunities here. So I've compiled a list of the ones I know about. I've sent them to Professor Murray, and he's going to send them out to all of you. I encourage you all to take a look at those. Both great opportunities for undergraduate research and grad school. And if anyone is interested in going out and finding their own tardigrades, there's something called the International Society of Tardigrade Hunters, which Thomas Boothby, the postdoc in our lab, runs this website, and it has instructions on how to go find them yourself. So that is my talk, and I will take questions.
Hi, my name is Lauren, and I actually have two questions. So the first question is, how long do tardigrades actually live under normal or ideal conditions? And then the second question is, where do you get your tardigrades for your experiments?
All right, so tardigrades, I think their lifespan is a couple of weeks. So much shorter than the amount of time that they can hibernate through. And the second question was where we get our tardigrades that we work with.
Yeah.
So you can buy tardigrades. There are a couple different companies that sell them. The cultures that we have, Thomas has been keeping for years, so I don't actually know where he originally got them. He brought them from the previous lab he worked in. But we've also just gone anywhere in North Carolina, any chunk of moss you pick up, if you throw that under a microscope with some water, you will find tardigrades. And that's probably true in Michigan too. I haven't found them in California yet, but I think California is a bit dry, so it's harder to find them.
So I've got a question. The mechanism of gelation, are you considering things like hydrogen bonding, electrostatic forces for those heads to come together? Is that what you're looking at?
Yeah, we think it's most likely hydrogen bonding from electrostatic interactions. We haven't gotten into the nitty-gritty of it quite yet. We've still been kind of fleshing out the big picture. But I think that's the most probable source of bonding. It's also possible that there could be some hydrophobic interactions, but I think that's less likely. I don't have evidence of it being less likely, though.
Dave Novak. Is there any evidence that protein that you're studying has any kind of allergic reaction or immune reaction in humans? And two, have you found or looked even for proteins that are similar to that in the human genome or DNA?
All right, so for the first question, that's something that we're currently working on funding to test. We want to take some of these proteins and inject them into mice and see if the mice have an immune response. We suspect that because they're intrinsically disordered, proteases in the bloodstream are hopefully going to chew them up quickly enough that there isn't time for the immune system to respond. And so that there isn't time for you to get sick or develop antibodies that might result in you having an adverse reaction in the future. That's all speculation though. So we're working on running that mouse study. It'll hopefully be happening. We're going to do a preliminary experiment at the end of October and a more thorough one once we are able to secure funding. What was the second question again?
Is there any do they sense there's a similar protein in humans anywhere?
Right. So there's nothing similar in humans in terms of sequence. There are some intrinsically disordered proteins that show up in humans. I don't think there's anything that there's nothing homologous, and there's nothing that I would really concretely say is similar besides being in the intrinsically disordered protein family.
And just a follow-up. So to your knowledge right now, there's no commercially available antibody to that protein.
That is correct. We're hoping that we might be able to get mice to make the antibody for us at some point. But if they are degraded very quickly in the bloodstream, that may end up being difficult. So that would be kind of a con of them not having an immune response. But
yeah, and not having any kind of stable structure, perhaps there wouldn't be an epitope for the antibody to
yeah, that's also potentially true as well.
So I'm wondering if any polymer chemist has approached you guys to use the proteins that you're working with here as a model to make, synthetic polymers.
Okay. Not yet.
Yeah.
We've been working with Sergei Shaiko's lab that really specialize. They're a polymer chemistry lab. They're the ones that taught us all of the rheology. So there's potential that they could take this somewhere. We'd certainly be open to additional collaborations, looking at that sort of thing. But for the moment, no.
Okay. Anybody else? Okay. Thank you very much.
Thank you for joining us.
Thank you.